This presentation is about the role of microorganisms, particularly bacteria, for their proteins and peptides for therapy and prevention of cancer and infectious diseases, both viral and parasitic. But we'll emphasize in this presentation how bacteria have been known to fight cancer. And this scenario has been developed over almost 120 years. So in one example, for example, we demonstrate that there are mice. These experiments can be done with mice bearing a tumor, as you can see on the flank, and a bacterium such as pseudomonas aeruginosa. And the question is, since pseudomonas aeruginosa is a pathogen, but it's fighting cancer in human, and it has the potential to fight cancer in human, is pathogenesis necessarily bad, or can pathogens actually be used in the therapeutic and preventive application of a problem disease such as cancer? And can our next generation drugs come from pathogens? So now we go back about 120 years to discuss the remarkable observations of Dr. William Coley. Dr. William Coley was a head and neck cancer surgeon at the Memorial Hospital in New York City, now known as Sloan Kettering Memorial Cancer Center. In 1892 and onwards, for several years, Dr. Coley actually found out that deliberate infection of his patients harboring tumors with bacteria such as streptococci, for example, allowed significant and sometimes total regression of cancer. He actually tried this out of 312 patients with sometimes inoperable cancers, and 190 patients showed complete regression of the tumors with a 62% success rate. His problem was, remember, this was 120 years ago, in 1892, 1893, 1894, around that time. So we are going back long time back, 120 years ago, when there were no petri dishes. He was a head and neck cancer surgeon. He was not a microbiologist, but he had that insight to find out that when some of his patients got infected with certain pathogens, streptococci for example, that the cancer in his patients would regress. But of course, a patient cannot have an infection forever because the infection would kill the patient. So he had to use, there were no antibiotics at that time, so he had to use sulfur drugs to get rid of the infection. And when he got rid of the infection of his patients, the cancer would come back. So that's the time when he made that observation, even though he was not a microbiologist, that bacteria, when they infect his patients, cancer patients, would actually allow regression of the cancer in his patients. And therefore, he said, why am I waiting? Why don't I isolate these bacteria from my patients that showed cancer regression and infect my otherwise healthy patients with cancer with these bacteria and see if the bacteria could actually allow regression of the tumors in his patients. And as mentioned, out of 312 patients, with sometimes inoperable cancers, 190 patients showed complete regression. But his problem was that the results were uneven and unpredictable because Dr. Cawley couldn't control the infection process. He had to inject, through intravenous injection, live bacteria into his patients, which of course allowed activation of the immune system. Some of these patients are old and very sick, and when their immune system got activated, they became sicker, and use of live bacteria created toxicity problems in such patients, sometimes resulting in the death of the patient. And of course, one's treatment modality must not kill the patient. So he demonstrated something very, very important and tangible, but it didn't take off at that time because he couldn't control the infection process. And you can look at the references cited here that actually describe many of his successes and some of these apparent failures. Since then, many people have studied the role of bacteria in fighting cancer. For example, again, you can look at the references. Salmonella and Clostridia, two bacteria, are known to target tumors for proliferation and tumor regression. So you can see on the table that when mice harboring B16F10 melanoma, these are skin cancers, when they had them, and if there was nothing else, only the vehicle, meaning that the bacteria were not there, the tumor doubled in volume in 2.74 days. So the tumors were growing rapidly, and they would become twice the size in 2.74 days. However, if the patient received 10 to the 4, about 10,000 colony-forming units of the bacteria, the doubling time for the cancer would go up from 2.74 to 4.32, meaning that the cancers started to grow slowly. So the doubling time went up. With 10 to the 5 CFU, it would be 6.06 days for the doubling of the tumor stalker. With 10 to the 6 bacterial colony-forming units, the doubling time would increase to 9.25 days. So that instantly tells us that the more bacteria entered into the body, the lesser was the growth of the tumor, so that the bacteria somehow were actually allowing the slowdown of the growth of the tumors. And when one injects these bacteria in mice without tumor, most of the bacteria would show up in the kidney and the liver. However, look what happens when the mice would harbor, let's say, a lung carcinoma, a lung tumor. 10,000 to 1 would be the ratio of the bacteria present in the tumor versus in the liver. Or in other words, the bacteria would target the tumor much more preferentially than the organs where they would normally end up with. And you can see those two references that actually discuss these applications in more detail. In 2001, a group from Johns Hopkins published this nice paper in PNAS, as you can see, and they demonstrated that chemotherapeutic and antivascular agents such as mitomycin C and dolastatin 10 allow the regression of a mouse colorectal and melanoma tumor. The tumors regressed much faster and in some cases completely vanished when spores of a toxin-deficient strain of Clostridium novae were introduced by IV injection. Some animals even remain tumor-free. However, 15 to 45% of the mice died. Look at this reference. Depending upon the size of the tumor, demonstrating the presence of toxic products. So you can see that in injection of live bacteria into tumor harboring mice would actually allow rapid regression of the tumor. But the problem would be that such bacteria would cause immense toxicity problem, and therefore if we are going to apply such live bacteria in the treatment of cancer in human beings, we have to address this toxicity problem and reduce it substantially so that using live bacteria would become feasible in the treatment of cancer in human beings. There is an important lesson here. People always thought physicians have great faith in immune system, whether in mice or in human. They always think that when one introduces live bacteria into the mice or even in human, and the cancer regresses, they say, oh, it's due to the activation of the immune system, and it's the immune system that's causing cancer regression. It is actually fairly well known that the immune system does protect either animals or human beings from cancer. But there was this interesting article by Hunter et al., as shown below. They showed that no effect of cytotoxic cells, macrophages, non-cytotoxic T cells, NK cells, etc., or major cytokines on tumor regression by Toxoplasma gondii, a parasite, was demonstrated in knockout and skin these mice harboring this B16F10 melanoma. They further demonstrated the production of infection-induced soluble factors that suppressed angiogenesis, meaning blood vessel formation, which is very important for cancer growth, in melanoma tumors. When the mice were infected with Toxoplasma gondii. But of course, they couldn't demonstrate the production of these soluble factors in tumor-bearing mice not infected with Toxoplasma gondii. So the conclusion was that Toxoplasma gondii, not a bacterium, but a parasite, the infection of Toxoplasma gondii will promote accumulation of soluble anti-angiogenic factor that allows tumor regression. So what did we learn? We learned that certain bacteria would allow cancer regression, although the mechanism of such regression is not understood or was not understood. And the last slide showed that even though people believed that bacterially induced regression of cancer, regression of tumors, was largely due to an activation of the immune system, the last slide showed that that wasn't necessarily so, at least in certain instances, as reported in that paper. It is at this time that we accidentally made some interesting observations, as shown in this slide. What we demonstrated was that some pathogenic bacteria, such as pseudomonas aeruginosa, that can infect the lungs of cystic fibrosis patients and can actually live in the human lungs, in this case the lungs of CF patients, for a long time, forming a thin layer of growth called the biofilm. So when these bacteria, pseudomonas aeruginosa in this case, lives in a very slow growing mode in the form of a biofilm on the lungs of CF patients, they know that they are going to be there for days, months, or sometimes years. And therefore, they consider the human body as their habitat. And because they were living in the lungs of CF patients in this particular instance, but they can also infect, Pseudomonas aeruginosa can also infect human beings under other conditions, such as when they are immunocompromised or when they get severely burnt and so on. They try to protect the human body, their habitat, when they are formed the biofilms, from outside invaders such as cancers, viruses, or parasites by producing protein weapons. One such weapon, as described here, is called Azurin, which demonstrate entry specificity in cancer cells. So if you look at the upper part of the figure, you would see that J774 is a mouse macrophage-like cell line, but it's actually derived from cancer tumor cell. So you can see that when azurin was labeled red with a dye and incubated with J774 type of cancerous cells, you can see that they entered in J774 cells. And the figure shows the blue dots are the nuclei stained with a dye called DAPI. So the nucleus is shown as blue sphere, but you can see surrounding the nucleus in the cytosol is the red colored azurin, meaning azurin has entered into the cell and is present in the surrounding cytosol, but you can also see that azurin has also entered the nucleus, which is labeled blue. However, instead of taking tumorous J774 cells, if you take peritoneal macrophages or mast cells and incubate these normal cells under exactly the same conditions and look for the entry of red-colored azurin protein, pseudomonas azurin protein, you would see only minute traces, nowhere as much as you can see in 6 hours, for example. But don't limit yourself only to mouse cells or tumors. Look at MCF7, which is a human breast cancer cell line, as opposed to MCF10F, which is a normal human breast cell line. When MCF7, the breast cancer cells, are incubated with azurin labeled with the red dye, you see the same thing. Lot of red colored azurin entered into the MCF7 cells and was present surrounding the nucleus in the cytosol, but you can see traces of azurin entering into the nucleus as well in 6 hours. But look at MCF10F, the non-tumor normal mouse breast cell, and you would see extremely low amount of azurin, if any, suggesting that azurin had entry specificity in cancer cells but not in normal cells. And when you see traces of azurin in the nucleus, you say, how does azurin get into the nucleus? And the next slide actually shows, see, there is P53. P53 is a tumor suppressor. It suppresses tumor formation. But for cancer cells to grow, they actually cause mutations in P53 so as to greatly lower its level so that it cannot suppress tumor growth by interfering in the cell cycle checkpoint as well as inducing apoptosis. So what azurin does, as we have shown, is that azurin forms a complex with tumor suppressor P53 in the domain where P53 interacts with another protein called MDM2 or HDM2, which ubiquitinates P53 and makes it proteolytically vulnerable, meaning that the P53 levels in normal cells are greatly lowered because of its ubiquitination by MDM2 or HDM2. When azurin interferes in the binding of MDM2 or HDM2 with P53, the ubiquitination is greatly reduced, so the azurin level significantly goes up, and then azurin causes apoptosis as well as cell cycle arrest, inhibition of tumor growth. But because azurin entered very, very preferentially to cancer cells and not to normal cells, such growth arrest as well as induction by apoptosis occurs primarily in cancer cells, but not in normal cells. The figure just below the P53 figure actually shows that azurin also inhibits angiogenesis, blood vessel formation. Blood vessel formation, angiogenesis, is very important for the cancer cells to grow rapidly to provide them with oxygen and nutrients. So azurin, by preventing angiogenesis in tumor cells where it entered, would significantly reduce the growth of the cancer cell. And therefore the question is, will azurin actually inhibit tumor growth in live mice, not in cell lines, but in live mice? And that's shown in the lower part on the left. What those two figures show is that when mice are injected subcutaneously with human tumors, such as breast cancer or melanoma, all the mice, let's say 20 mice, we take 20 mice, give all the mice subcutaneous injection with human breast cancer, MCEP7, or melanoma, so that these tumors will grow in all the 20 mice. Now we divide these mice into two groups of 10 mice each. One group of 10 mice, we give an injection of saline, so that's called the control. In the other group of 10 mice, we give an injection of azurin, 1 milligram azurin per injection. So 10 mice get only saline, while 10 mice, all harboring tumors, will get 1 milligram azurin dissolved in saline. And then we follow the size of the tumor as the tumor grows in these mice. And those are shown in the graphs below. And what the graphs actually show is that azurin can cause shrinkage of the tumor anywhere from 58% to 85%. So azurin actually appears to allow tumor shrinkage in live mice, and the histological examinations of the tissues of these mice demonstrate that they look pretty normal, suggesting that these mice, where the tumors shrank after azurin treatment, demonstrated no obvious histological pathologies, and therefore presumably azurin is not very toxic in these mice. But is there any real evidence that azurin reduces tumor or fights cancer metastasis in human? Here is a letter I got from a physician in Fairbanks, Alaska. We mentioned earlier that live bacteria can cause tumor shrinkage in mice when injected. And these bacteria, remember these experiments were done in mice. These bacteria are basically Salmonella, Clostridia, Listeria, Microbial, Bovis BCG, and so on. But not previously reported is any role of the bacteria Pseudomonas aeruginosa that I mentioned previously produces this weapon, routine weapon azurin, that very preferentially enters into cancer cells and causes cancer growth inhibition through multiple mechanisms, including stabilization of tumor suppressor P53 through complex formation, inhibition of angiogenesis, and other mechanisms that we wouldn't talk into. But the question we are asking is, that's okay. But is there any evidence that Pseudomonas aeruginosa can fight cancer, tumors in human patients? So here's the letter. So I got this letter from a physician in Fairbanks, Alaska. The name and the affiliation is, they're shown in the bottom. And the physician said, dear Mr. Chakrabarty, a patient with high-grade prostate cancer near the ureterovesicular junction has been under my care for the past year. He has been stable and relatively pain-free until 3 weeks ago, when we were finally able to eliminate a year-long catheter-induced pseudomonas infection at the site of the plastic ureteral stent tumor site. Post-surgical MRIs, magnetic resonance imaging, now reveal metastasis to pelvic and hip bones, liver, and lungs. A crazy idea, so I thought, occurred to me. Is it possible that a persistent pseudomonas infection at the tumor site could have somehow kept the cancer in check? A search on the internet led me to your work. I am writing to you because this previously stable patient is now deteriorating rapidly, and I thought you would be able to tell me whether there is any treatment, drug or trial, based on pseudomonas that might give him a chance to slow the cancer. Did you see what she said? This is a clinical observation that if there is a tumor, in this case a prostate tumor in a prostate cancer patient, and if there is a stent, a catheter, inserted near the tumor for other reasons, and if the catheter gets infected with a biofilm of Pseudomonas aeruginosa, the patient would do well. The cancer would remain in check so long as the Pseudomonas is there. But physicians don't like bacteria in the body. particularly pseudomonas aeruginosa. So the physicians used a lot of antibiotics to get rid of the pseudomonas, and they were ultimately able to get rid of the pseudomonas, and look what happened. The cancer spread. It metastasized almost immediately within 3 weeks, as she said, suggesting, as she thought, that the pseudomonas was fighting the growth and the metastatic migration of that prostate cancer. An interesting clinical observation. Let me now tell you a little bit about how cancer is treated now. So basically, cancers are treated by small molecule drugs and or monoclonal antibodies with a single target site or a very limited number, but the targets are very, very similar. This is done by the pharmaceutical industry through a process called rational drug design, whereby they choose what's called a target, and they would use thousands of chemicals to find out one in a high throughput screening that would strongly inhibit that target. And because the target is important in the growth of the cancer, if there is a strong inhibitor of that target, the cancer growth would be significantly reduced. So that strong inhibitor would become potentially a drug if it doesn't demonstrate toxicity or other ill effects and efficacy in subsequent preclinical and human phase 1, 2, and 3 clinical trials. So the drugs on the left that you are looking at, both small molecule and monoclonal antibodies at the bottom, have been developed by rational drug design. The targets are shown, and you can see that the targets are very limited. The cancers are shown. CML is chronic myelogenous leukemia, GST is gastrointestinal stromal tumor, RCC is renal cell carcinoma, hepatocellular carcinoma, and so on. But look at the side effects, and the references are there. All these single or limited targeting, either small molecule or monoclonal antibody drugs, have high toxicity side effects, making chemotherapy that employs these drugs for the treatment of cancer a very toxic treatment modality. The reason being that the target, while they are often hyperexpressed in cancer cells, is also present in the normal cells. So, if you strongly inhibit a single target, you get two problems. One is toxicity, because over a period of time, because it's a strong inhibitor, eventually it will start inhibiting the same targets that are present in less amounts in normal cells, particularly somewhat rapid growing normal cells, such as hair follicles, for example, or the cells that line up our stomach, because stomach is full of acids, so they got to regenerate themselves quickly. So these faster growing normal cells also become target of the chemotherapeutic drug, so that major side effects are the problems in chemotherapy. And you can instantly see that an ideal drug therefore would be one that would very preferentially enter into cancer cells and not to normal cells, so as not to upset the metabolism of the normal cells, but should also inhibit, not very strongly, not very stringently, somewhat modestly, but multiple sites so that the cancer cells cannot become resistant too quickly as they do for chemotherapeutic drugs with a single site. If a drug attacks only a single site, the cancer cell can change that site or shut it off. Or if it's a small molecule, cancer cells can activate their efflux pump to pump out the small molecule drugs. They can do it if the drug specifically entered into the cancer cell and then stop the cancer cell growth by interfering in multiple steps at a modest rate so that the cancer cells wouldn't really know which step to change or shut off because there are multiple steps and the inhibition is modest, but cumulatively as a whole the inhibition is strong enough to inhibit cancer cell growth. I mentioned about azurin produced by pseudomonas aeruginosa. I would also like to mention that pseudomonas aeruginosa has a sensory mechanism to feel the presence of the enemy such as cancer and send its weapon azurin outside to fight cancer. And how do I know that? We have actually published a paper where we demonstrated that if one grows pseudomonas aeruginosa in a growth medium, azurin is in the periplasm. So one wouldn't see much of azurin in the growth medium. If one adds some human cancer cells, such as breast cancer MCF7 cells, for example, instantly azurin will come out of pseudomonas aeruginosa in the growth medium and will enter into the cancer cell, but of course not to the normal cells fighting cancer. The fact that pseudomonas instantly releases azurin when it faces a cancer cell shows that pseudomonas can recognize its enemy and sends its weapon out to fight the enemy. That's one interesting feature of the bacterial anti-cancer protein weapon. But there are other interesting features as well. For example, we were interested in finding out how many other pathogenic bacteria would produce azurin-like proteins. And we took the azurin sequence and checked the sequences of many other bacteria, and we found out that among others, there is one bacterium, neisseria meningitidis, that also produces an azurin-like protein. But very interestingly, when we looked at the sequence, we found out that there were 39 extra amino acids in the N-terminal of neisseria azurin called LAZ. L-A-Z, LAZ means lipidated azurin. The neisseria azurin is lipidated and has been known much before we found all these things and used to be called LAZ. Okay. So we became interested in knowing why neisseria would specifically put an additional armor, a 39 amino acid epitope in the N-terminal of its weapon azurin, because neisseria azurin had significant sequence identity with pseudomonas aeruginosa azurin, except it had a 39 amino acid epitope called an H8 epitope. It occurred to us that neisseria meningitidis causes meningitis. Meningitis is an inflammation of the brain membrane meninges. That means that neisseria can cross the blood-brain barrier to go to the brain that pseudomonas aeruginosa is not very well known to do. So you can see uh, on the left, you can see that when one uses, again the same thing, azurin, there is also another similar protein belonging to the copper containing proteins called leucocyanin, called cupridoxine. They all belong to this group called cupridoxine. You can see when you use the human cancer cell line, melanoma cell line MEL2, both azurin and leucocyanin labeled with a red dye entered the cell surrounding the blue labeled nucleus. MCF7, the human breast cancer cell, the same thing. But when both of them were used in MCF10A1, which is a normal breast cell line, say azurin entered very inefficiently in the normal cell, but leucocyanin entered almost as well, suggesting that while azurin has entry specificity in cancer cells and doesn't enter into normal cells very much, leucocyanin does, another cupridoxine. So the question was, this H8 epitope of 39 amino acid, why was it introduced in the N-terminal end of azurin by neisseria during its evolutionary step? Those data are shown in the right-hand side. So we assumed that neisserial azurin had this additional armor, H8 epitope, to allow it to enter into a brain tumor if neisseria resides during meningitis in the brain meninges, and there is a brain tumor that it has to fight. And indeed, if you look at the cytotoxicity of pseudomonas azurin, LAS, and we also cloned the H8 epitope from the N-terminal end of LAS and put it in the N-terminal end of pseudomonas aeruginosa, and now we labeled it H8P azurin. And look at the cytotoxicity. What you will see is that pseudomonas aeruginosa has much lower cytotoxicity for glioblastoma or astrocytomas, two brain tumors, than either LAS or H8 azurin, suggesting, particularly for H8 azurin, that the introduction of the H8 epitope in the N-terminal of pseudomonas aeruginosa confers on it the ability to fight brain tumor much more effectively than azurin will. Then if you look at the entry of azurin in human breast cancer cell line and the glioblastoma cell line termed LN229, look at LN229 entry. Red labeled azurin enters very little, suggesting that there is an entry barrier for azurin to enter into a brain tumor, but either H8 azurin or LAS will enter much more effectively, suggesting that the H8 azurin is allowing azurin to enter into glioblastoma cells and exert its efficacy as an anti-cancer agent. But look at also the entry of azurin, H8 azurin, and LAS in MCF7 breast cancer cells. There, azurin and H8 azurin are basically very similar, suggesting that the H8 moiety is particularly effective when the tumor is a brain tumor as opposed to being a breast tumor. And this is finally shown in the lowest bottom slide, when azurin, H8 azurin and LAS are labeled with a green fluorescent dye so that they all label green, and these purified green fluorescent proteins are injected into the peritoneum of the mice so that they will go to the blood, and later the brains of these mice after 24 or 48 hours are isolated and checked for the entry of the green fluorescent proteins, azurin, H8 azurin and LAS. You can see that very little azurin, green fluorescent azurin, is present in the brain, suggesting that azurin had problem getting from the bloodstream to the brain. Simply putting the H8 epitope in the N-terminal part of azurin allowed a lot more azurin and also LAS to enter the brain, suggesting that the H8 epitope was facilitating the entry of azurin from the bloodstream into the brain. This then raises an interesting question. There are many neurological diseases that we face. Parkinson's is one, Alzheimer's is another, and part of the problem in treating these neurological diseases is the lack of effective drugs, because very few drugs will actually cross the blood-brain barrier to enter, for example, to attack and remove the plaques formed in Alzheimer's patients' neurons, brains. Can H8 help in facilitating the entry of any potential drug from the bloodstream to the brain? So that's an interesting question that can be addressed apart from the use of H8 azurin in treating brain tumors. This slide simply shows that azurin can not only allow shrinkage of tumors through complex formation with p53, reducing angiogenesis by forming a complex with vascular endothelial growth factor and so on, but it can also greatly reduce the migration of certain breast cancer cell lines that overexpress a particular protein called p-cadherin. So this basically shows, if you look at the right hand side, that p-cadherin is a poor prognosis factor for highly invasive breast cancers, and azurin, the multifunctional therapeutic protein, can potentially not only inhibit angiogenesis and induce apoptosis and inhibit proliferation through stabilization of tumor suppressor p53, but can also potentially prevent invasion of cancers. And in this slide, what it shows is that azurin, unlike those single targeting drugs of the pharmaceutical industry, azurin can actually act at multiple states as demonstrated by this transcriptomic array-based gene expression profile. On the left are upregulated genes when the cells such as MCF7AZ or MCF7AZ hyperexpressing p-cadherin are treated with azurin. You can see the number of genes that are hyperexpressed, and these genes are involved basically in transport, membrane organization, induction of apoptosis, all very useful to prevent cancer growth. And in the bottom shows down regulation of genes that promote cancer growth. So those genes that are down regulated by azurin are those that normally would promote cancer growth, suggesting that azurin is a multi-targeting drug, specifically attacking various steps in cancer growth. One of the interesting subject about azurin is that it not only attacks cancer, but it also appears to attack the malarial parasite named Plasmodium falciparum. And how can azurin do it? Because azurin, if you look at the left, has structural similarity with a protein called MSP, merozoite surface protein 1, and a part of it called MSP119. And the KD values of the interaction of azurin and LAS with this protein are shown at the bottom of the slide. And then on the right hand side, what it shows is the parasitemia and the concentration of azurin. So if azurin concentration is increased during infection of RBC by the parasite Plasmodium falciparum, and then we look at total number of parasites called parasitemia, you can see that there is steady decrease with higher concentrations of azurin, H8 azurin, and LAS, suggesting that these proteins appear to have growth inhibitory activity against the malarial parasite Plasmodium falciparum. In the previous slide, we showed that azurin not only has structural similarity with MSP119, but it also appeared to have structural similarity with CD4. CD4, of course, is the receptor of the virus HIV-1 that causes AIDS. So we tried to find out whether that means if azurin will interfere in the binding of the HIV/AIDS virus HIV-1 with CD4. And those data are shown. These are the surface plasmon resonance data demonstrating KD values, and below is the inhibition of viral growth with or without treatment with azurin, H8 azurin, and LAS. And you can see azurin, H8 azurin, and LAS all will significantly 75 to 80% or even higher inhibit the growth of HIV-1 virus. So you can instantly see that azurin appears not to have anti-cancer activity, but also appears to have antimalarial and anti-HIV-1 activity. As mentioned before, current industry trend is one target, one drug, as we saw for cancer. But look at azurin. One protein targeting many diseases, cancer, HIV/AIDS virus, HIV-1, and malaria. And of course, as you know, that AIDS patients get not only HIV-1 virus, but because of the lowered immunity, they often develop cancers, Kaposi sarcoma, for example, or get infected with various bacteria or parasites such as the malarial parasite Plasmodium falciparum or other similar parasites. So the point is that azurin therefore could potentially be effective not only in the treatment of the AIDS patients, but also in the treatment of AIDS patients who have been infected with malaria or who might have developed Kaposi sarcoma and so on. One drug, one protein drug, multiple targets. Such a drug doesn't exist. But of course that means that one would have to show that azurin is non-toxic in human, which we haven't shown as yet. But azurin has other properties that are very, very interesting. For example, I talked about the therapeutic effect of azurin, meaning that if there is a tumor, azurin will allow shrinkage of the tumor. That's a therapeutic activity. But azurin can potentially prevent the onset of cancer. And how do I know that? Look at the data. So this is an experiment where one can grow normal mouse mammary cells. Normal cells grow rapidly, but they can be grown in culture. And then one could add a potent carcinogen such as DMBA, dimethyl benzanthracene, a potent carcinogen. So if in that assay where normal mouse mammary cells are being grown, if one adds certain concentrations of DMBA, the carcinogen, as shown in the left, presence of DMBA will produce what's called precancerous lesions. These are shown in the left-hand slide. These precancerous lesions have the potential to become cancers, full-fledged cancers. So in this assay system, where normal mouse mammary cells are being grown and one added DMBA to look at the number of precancerous lesions that are formed, if one adds different amounts of azurin shown in group number 6 or a fragment of azurin shown here as P28, this is a 28 amino acid fragment of azurin, a peptide of 28 amino acid from azurin amino acid 50 to azurin amino acid 77. You can see that both azurin and P28 will demonstrate significant inhibition, 60, 70, 75% inhibition of precancerous lesion formation. This is prevention of the onset of cancer, meaning that when a normal cell becomes cancerous, azurin doesn't enter into normal cells, but will instantly enter into the cancer cell and kill it before it becomes a full-fledged adenoma or carcinoma. That's prevention of cancer by azurin in addition to its therapeutic effect. Question: can azurin be used as a drug in human cancer patients? This is a question we asked by forming a company named CDG Therapeutics, Inc., www.cdgti.com. And they raised about $12 million. And the National Cancer Institute of the United States government was very generous in performing the preclinical trial, not with azurin, because as a protein, azurin will have to be isolated from bacteria, and therefore it might be contaminated with the LPS, and therefore it goes through a lot more stringent regulation for clinical application as recommended by the US Food and Drug Administration. So it's always easier not to use a protein, but to use a peptide that can be chemically synthesized and therefore is labeled as a drug because there would be no cellular contaminant. So when the company approached the FDA to conduct phase 1 human clinical trial, after the NCI demonstrated that this peptide P28 had no toxicity in animals, including primates such as monkeys, the FDA said, okay, go ahead, but there are ground rules. Because one doesn't know if P28 will have toxicity in humans, one would have to use a very low concentration initially and go higher up if there are no side effects. So the company had to try the effect of P28 in phase 1 clinical trial in 15 patients, 11 male, 4 female, age range 50 to 80. Those 15 patients had different cancers. 7 had melanoma, 4 had colon, 2 had sarcoma, 1 had pancreatic cancer, and 1 had prostate cancer. The treatment modality was IV bolus, so IV injection, 3 times a week for 4 weeks, followed by 2 weeks of observation before the next higher dose. And the doses were, the lowest dose was 0.83 milligram of P28 per kg body weight of the patient. So one recruits 3 patients. One gives 0.83 milligram per kg body weight in these 3 patients. And if there are no side effects, if one recruits 3 more patients and give all the 6 patients the next higher dose, 1.66 milligram, and so on, until all the 15 patients are given the highest dose, assuming that there is no side effect. And there was none. According to the physicians, there was no adverse side effect that could be attributed to P28. No immunogenicity either. At very low dosages, there was not much effect, but at medium dosage, the disease became stable, meaning that the cancers didn't increase any further. 2 patients had partial regression of their tumor, 1 prostate and 1 melanoma patient, while 2 patients had complete regression of their tumors. I would emphasize here that all the 15 patients were stage 4 cancer patients, meaning that they had multiple solid tumors. The tumors were not responding to any conventional drugs, and the patients had a life expectancy of 6 months or less. The fact that 2 patients had complete regression of their tumors and 2 patients had partial regression of their tumors, but the tumors were not responding to any conventional drug, tells one that P28 is a very unique drug. It's not like the kind of drug that the pharmaceutical industry comes up with. It's a bacterial protein weapon drug that we accidentally found out. And that raises an important question as to how many other bacteria can produce similar protein weapons that would fight cancer or some of the agents of infectious disease. The fact that P28 has cancer preventive activity has some relevance to what's happening right now. For example, even though I'm not going to discuss patent cases for this talk, I would simply demonstrate to you that one can get cancer by basically three ways. Cancer is nothing but an accumulation of mutations in the human genome. The minimum number of important mutations called driver mutations is about 4 or 5. P53 mutation, as I mentioned before, is one such mutation. 50% of cancer patients demonstrate mutations in P53. There are others, as you will show in a few minutes, particularly for animals such as mice. So one can get these mutations. It could vary anywhere from 4 or 5 to 10 or 15 or 20, 25, or even 50 or 100. So key mutations that are primarily responsible are called driver mutations, while the other mutations that add to the rapid proliferation of the cancers are called passenger mutations. So older people are much more susceptible to cancer because they accumulate mutations in their genome as they age. One could also get cancer if one has a bad lifestyle, such as smokers, for example, are susceptible to lung cancer, or people who lay under the sun for long periods of time become susceptible to developing melanoma. So, environmental mutagens and carcinogens can accelerate the onset of cancer. But there is another way people can get cancer, and that is through genetic inheritance. One could get some of those mutations from their parents. And the critical example is that of women in particular, but men also get them. Certain mutations called breast cancer, BRCA1 and BRCA2 mutations in those two genes in the human genome, into different chromosomes. So men with BRCA1, BRCA2 mutations become susceptible to breast or prostate cancer, but the frequency is not very high. Women, however, can become very susceptible if they inherit BRCA1 or BRCA2 mutations from their parents to breast and ovarian cancer. And this company, Myriad Genetics in Salt Lake City, Utah, had 7 patents that covered the genetic screening of breast or ovarian cancers in women with a family history of BRCA1 and BRCA2 gene mutations. So the case was whether human genes and mutations such as BRCA1 and BRCA2, for which Myriad Genetics had 7 patents, are patentable or not. And the Supreme Court on June 13th in 2013, in a unanimous decision said they are not, but cDNAs are patentable. That's a different topic that I normally talk about in meetings in front of intellectual property lawyers and other people interested in patent laws. But the question here is, what happens when young women of childbearing age, let's say a girl of 20, 25, 30, 35 years age, with a family history of breast and ovarian cancer, they go to the physician, and the physician will tell them that you have a family history of breast or ovarian cancer, you should get yourself checked for the presence of BRCA1 or BRCA2 gene mutations, and they would go to Myriad Genetics, and Myriad Genetics would take blood samples and determine if they have the mutations, and in some women the mutations should be present, and in others the mutations should be absent. But the important thing is when a young woman of childbearing age with a family history of breast or ovarian cancer will go to Myriad Genetics and get herself checked, and Myriad will tell her that yes, she does have the mutations, and she will come back home and she will say, what do I do now? There's an interesting story about an actress who decided, because she had a family history of breast cancer, to have radical mastectomy, surgical removal of the target organs such as breasts and ovary. But you realize that for a young woman of childbearing age, that's such a traumatic thing to do because she will never have any children. She will be disfigured for the rest of her life, and that's a terrible, terrible, decision to make. Because P28 has been shown to have both therapeutic activity without any toxicity at the dosage that was given to those 15 stage 4 cancer patients for several months. The question is, can P28 work to prevent cancer in such vulnerable people? While it's not easier to demonstrate therapeutic activity as opposed to preventive activity, because how does one show cancer prevention in people who don't yet have cancer? How long must the people will wait? What would be the mode of introduction of P28 in such people? How often? Things of that sort. There are many, many important decisions that need to be made in this context. But for this presentation, I could simply mention to you that there are ways by which we can determine if P28, ideally Azurin, when Azurin has gone through preclinical and phase 1 clinical trial to demonstrate not to have any toxicity. But right now that has been done with P28. So let's talk about P28. How can we show that P28 has cancer preventive activity, at least in mice? So it's known that one can introduce lung cancer in mice if one can introduce two activated oncogenes, P53 and KRAS, using lentiviral vectors. So that's shown in this slide. So the wild type, when they are given as lentiviral vector to oncogenes, KRAS and P53, they will develop lung cancer. And the question is, under such scenario, if we take the mice with the KRAS and P53 activated oncogenes in lentiviral vectors introduced into their genome and allow the mice to inhale P20 or Azurin, let's talk about P28 right now because it has completed phase 1. Will P28 be able to prevent cancer in such mice that have become very, very vulnerable to developing lung cancer because of the introduction of these two activated oncogenes? Actually, another group at Salk Institute has shown that they can produce glioblastomas, brain tumors, if they would inject mice with 3 such mutations, AKT, HRAS, and P53. And one could do the same kind of experiment by introducing at the same time The azurin gene or the P28 peptide as a fusion protein using lentiviral vector allow them to be expressed at the same time and see if simultaneous expression of azurin or P28 gene along with the activated oncogene will prevent the number of tumors in such mice. So those are for the future. I will stop here by simply mentioning to you that azurin or P28 are not the only protein or peptide with anti-cancer activity. There are other bacterial proteins such as arginine deaminase. We talked about last. We have demonstrated that there is another protein called MPT63 in Mycobacterium tuberculosis or Mycobacterium bovis with anti-cancer activity. Raise the question as to how many pathogenic bacteria with long-term habitat in human tissues such as Microbacteria or Pseudomonas aeruginosa, and there are others, will produce the protein weapons to fight cancer, maybe viruses, maybe parasites, to protect their habitat. That's the most exciting project that I think microbiologists as well as bacteriologists will have to deal with in the near future. Thank you, and I hope if you have any questions, you will send me an email and I'll be happy to respond. Thank you.